I'm going to give a seminar trying to describe what we can do with soft X-ray tomography, because that's not a very known technique, and I hope I will convince you that it is useful for many different research. So to start, this is a first slide to try to put cryo-soft X-ray tomography in the framework of biological techniques today. As you all know, imaging across resolution scales is essential to understand your biological problem. And from the left side of my slide, which is the lowest resolution techniques, but which are tackling larger sample sizes, and to the right side of my slide, which are the highest resolution techniques, but are tackling smaller samples. Cryo-soft X-ray tomography is sitting around 30 nanometer resolution half pitch with a sample thickness of a few microns up to 10 microns. So what is a transmission X-ray microscope? Well, it looks like any of other microscope you have probably used in your life. So the source in this case in synchrotrons, it's far away, about 25 to 26 meters away from the microscope. We have a capillary lens and an objective lens, which will set your resolution. The sample is sitting on the focal planes of the condenser and the objective. The collection of the data is pretty fast. We are doing plus minus 70 degrees in few minutes of data collection. We have a natural contrast because we are working in an energy range which is called the water window, and this is because you can see it on my right bottom plot. The water is transparent, so the oxygen is not absorbing, and the carbon is highly absorbing. So this means that we do not need to section our samples, that we can go through the full cell without sectioning and without staining. So what we are collecting is the transmission signal, and when you reconstruct your tomography, due to the Beer-Lambert law, the intensity that is transmitted is proportional to your linear absorption coefficient. So when you reconstruct the tomography, each of your voxel, the value of each of your voxel, is directly the linear absorption coefficient of your material, so of your atoms. Because of this, by tuning also the energy, you will be able to do spectroscopy in your sample. So this is a typical slice, reconstruction slice that you can have. You can see the mitochondria, the ER between the mitochondria, and at the bottom left you have the nucleus and the nuclear membrane. The gray values here are directly the absorption coefficient, the line absorption coefficient. So vacuoles are appearing whiter than lipid droplets, for instance, that are appearing darker in the image. So which is the workflow that we are following? We are using electron microscopic grids to grow the cells on top or to deposit cells if the cells are in suspension. We are using, in general, find the grids, so this makes it easier to find in different instruments the same cell, the same interesting cell. And you have a sketch here of how the workflow goes. We are looking at these cells growing in vivo, then we vitrify the grid. We can again look at the fluorescent signal if you're using fluorescent tags. In cryo, after vitrification, the grids are screened mainly to look at the at the number of cells, of interesting cells that you have in your grid, and you can also locate them for going into the X-ray imaging microscope afterwards. We load the grid onto the grid holder and then into the microscope chamber, which is at UHV vacuum. So from vitrification, of course, to X-ray imaging, everything is done at cryo temperatures. This is again the same workflow, but with some images now. So we have a visual inspection of the grid. This is my top left image. Then you can do a fluorescence map. This is before loading the grid into the microscope. Then we can load the grid into the microscope, where we have again a fluorescent microscope, visible light epifluorescence, so that we can again find in the e-image here the interesting cell, which is labeled with this yellow square. If you look in the X-rays, this is what you have, the D image, where you have a cell in this square. And on the right, you have the overlay of the X-ray image and the fluorescent image. In this case, this sample was a cell infected with rotavirus. And the viroplasm of the rotavirus, was expressing GFP. So the viroplasm appears in green signal, and the mitochondria were labeled with mitotrack. So after that, after choosing your field of view, so the region where you will do the tomogram, the point here is that the cells are in general bigger than the field of view that we have in the microscope, which is between 10 and 15, 16 microns. So because this field of view is smaller, you use these markers, these fluorescent markers, to realize which are the regions of interest that you need to image. And then after that, you will collect your tomogram from -70 to 70 degrees. And this is how the raw data looks like. These are non-normalized projections. You have the nucleus on the right with the two nucleoli, the nuclear membrane and the cytoplasm. These are 141 projections, and they are collected in a few minutes. So what can we see from our reconstructed volumes? So in this case, I'm showing some fibroblast cells. You have on the upper left corner one of the grid meshes where you can see 4 cells. The yellow square again shows the region on the right upper corner, which is the reconstructed volume, where you can see mitochondria, lipid droplets, the ER, plasma membrane, the nucleus on the upper corner. Then on the C image here, you have again the nucleus with the double nuclear membrane, the pores, the nucleoli, nuclear channels. On the right bottom corner, you can see that there is blebbing of the nuclear membrane. The heterochromatin, because it's more packed than the euchromatin, will appear denser than the rest, than the euchromatin. And yeah, this is the typical images we are having. Again, mitochondria, ER between the mitochondria. On the right image, you can see the Golgi and lipid vacuoles, some more ER, mitochondria again. And on the right image, again, you have a nuclear pore on the nuclear membrane. So this is a volume. Yeah, it's working. The movie is working, yes. So we are going through the volume of the cell, the reconstructed volume of the cell, again with the mitochondria, the ER behind, involving, let's say, around the mitochondria. So this is the typical volumes that we are acquiring with soft X-ray tomography. So now if we go at which is the resolution that we can achieve, it's between 25 to 30 nanometer half pitch. And this is set by the lens, and this is what I'm putting here, which are the limits of the technique. The lens is giving us the resolution, but also the depth of field that we can get. The higher the resolution, the lowest the depth of field. So soft X-ray tomography normally is using two types of lenses, 25 and 40 nanometers. So that with the 25 nanometer, we are getting about 1.3, 1.2 microns of depth of field. And with the 40 nanometer, we are getting 3 to 3.3 microns of depth of field. In addition to that, to these limitations, we have the missing wedge. Because we are using flat samples, we cannot rotate plus minus 90 degrees. We are rotating only plus minus 70 or plus minus 65, depending on the lens that we are using. So this means that, as you can see, you have a missing wedge. And this missing wedge is visualized in your volumes, because when you have, for instance, on the F image on the bottom left corner of my slide, these mitochondria here, the cristae, are not well visible because they are parallel to the, they are perpendicular to the axis of rotation. So some structures will be hidden due to this missing wedge. And the way to recover a little bit of this missing information is doing what we call dual tilt, which means that you have first your sample at an orientation, you do the tomogram, and then you rotate it 90 degrees and you do the tomogram again. This will make that, as you can see in the sketch on the right, that instead of having a missing wedge, we have what we call a missing pyramid, but the missing information will be smaller, so that, for instance, at 90 degrees, the cristae of that mitochondria that we couldn't resolve first are well resolved. And then if you combine everything, if you combine the two tilt series, the missing information that you'd had in the first one will be recovered by the second one. And of course, having the structure with these tomograms will not be enough to understand what is going on. And we generally work in with correlative imaging strategies. And this can be using visible light fluorescence, of course, or also other techniques such as hard X-ray fluorescence, for instance, or even you can combine things with electron microscopy. So let's go into two examples that I have chosen today to show what is possible. The first one is dealing with understanding pathogen infections. In this case, Javier Conesa, who was a postdoc at the Beamline until last year, he was interested in looking and understanding, the asexual life of Babesia, which is a parasite that is infecting vertebrates, not only wild, but also domestic animals and also humans. And this parasite has a very complex asexual life cycle with a lot of different stages inside the erythrocyte. And Javier was combining confocal microscopy and soft X-ray tomography because confocal was giving him the chronological order, let's say, the chronology of this life cycle, while soft X-ray tomography was giving him the structure of the parasite inside the red blood cell. So by combining both of them, he was able to describe the full cycle of this parasite. And for that, he collected 218 tomograms. And this is again because the technique is quite fast and allows you to image a lot of different cells in a few days of beam time. And again, we are following here first invisible light microscopy, the fluorescence of the mitotracker, which was red, which was indicating that the red blood cell was infected. So we have on the right in the X-ray mosaic the cell of interest, and this on the bottom left you have a a virtual reconstruction or slice of your volume, and the segmentation on the bottom right of the different parasites inside the red blood cell. So this parasite has 7 main intraerythrocytic stages, and you can find them in this picture here, in this figure. The merozoite is the one that is with the parasite before infecting, before entering the red blood cell. And then from the merozoite, you go to the tropozoite, double tropozoite, parapiriform, the double parapiriform, the tetramaltese cross, and the quadruple tropozoite. And close to these segmentations that you can see where each color is showing a different structure in the cell, the plasma membrane of the parasite is in yellow, the dense granules are segmented in red. The mitochondrion is segmented in light blue, apicoplast in violet, and the nucleus in dark blue. You have some insets which are showing the Giemsa staining and also the confocal image of that stage. But in addition to these 7 main stages, this parasite has a life cycle where it goes from budding to fission to budding again in a very complex manner. And in addition to these 7 main stages, you have a lot of transient stages that could be described thanks to chronologically the confocal and to the soft X-ray tomography when you were finding each of these stages in the different cells. So as a little summary of this part, I just wanted to remember you that we have a resolution of 25 to 30 nanometers, and these allow us to look at cellular alterations that can be due to pathogen infections or can be due to diseases or to any other type of things, such as, for instance, drug treatment. And that, of course, understanding pathogens, the pathogen cycle in host cells is important for designing efficient and fast strategies to prevent diseases. Now I wanted to show you another example, which is biomineralization, because I wanted to show you something which has to deal with spectroscopy as well as tomography. So in this case, our collaborators from the Weizmann Institute, which come from Keren, which is the PhD student of Professor Lia Haddadi, she's interested in the calcium transport in sea urchin larvae. And by tuning the energy of the way, by tuning the energy, at the beamline to the calcium L2 edge, you are able to get a differential absorption between what is calcium and what is not calcium. And this is what is shown in the upper row here. The left picture is the calcium, the image at the calcium L2 edge. The middle one is the image before the edge, and the third image is the difference in absorption between the first image and the second image. And then if you do tomography, then you have the reconstruction of the calcium vesicles that you have inside these cells. These are primary mesenchymal cells, which are the ones that are taking seawater by endocytosis and concentrating the calcium in a network in a network of vesicles. And so by studying each of these vesicle particles, you can know which is the chemical species that you have, which is how is the calcium, which is the calcium species. So on the left side, you have plots, different plots of what you will get if you have seawater, what you will get if you have hydrated amorphous calcium carbonate, anhydrous amorphous calcium carbonate, and calcite. So by looking at the ratio of the peaks, of the L2 peaks, pre-peak and the peak, you can know which is the phase of each of the vesicles. So by doing this in 5 different cells and looking and analyzing 700 calcium-containing vesicles of these 5 primary mesenchymal cells, what you could see is that you have a continuum disordered phase from seawater to calcite, but most of your calcium-containing vesicles are between the seawater level and the hydrated amorphous calcium carbonate, and the other half are from from the hydrated amorphous calcium carbonate to the anhydrous calcium carbonate. So with this, what I wanted to tell you is, in addition to get some the structure within the cell, you can also have, by differential absorption or by directly spectroscopy, so the full plot, the full spectra, you can tackle which are the chemical species that you have in your cell. Now I'm going to show you a last video where which wants to summarize a little bit what does it mean imaging across scales. So you have fluorescent microscopy, you have your grid, and you have your interesting signal that you have tagged with the fluorophores, and you choose the corresponding cell. This is X-rays. You have the region that you're interested in, and you will do the tomography, the soft X-ray microscopy tomogram, where you have mitochondria and then the viroplasm. Now we are getting inside the volume, and what you can see here, these are the viruses containing the ER. Okay, the viroplasm is here, and the viruses are budding from the viroplasm to the ER. This is, this is electron tomography. So we are combining electron tomography from thin epoxy resin for higher resolution. And you will see that you have mature virions and which are enveloped viral particles. And of course, you could go to single particle and put your model of your rotavirus within the full frame. of this imaging across scales. So finally, my take-home message is that cryoscopic X-ray tomography can be useful to understand cellular organization during pathogen infections or diseases, or even when you have an infection, to evaluate the cellular recovery once you have used a certain type of specific drug treatment. It is always combined with, in general, fluorescence, visible light fluorescence, but can also be combined with hard X-ray fluorescence. I haven't shown this here today, but this is feasible. We can not only do tomography, so 3D reconstruction, but we can also use the energy tuning to do spectroscopy of your cell. And many of our users and ourselves are always working in correlative imaging strategies to understand what is going on inside of the cell. And finally, and before thanking you for your attention, just to remember you that 2 of the beamlines that are in this iNEXT-Discovery consortium are giving access to soft X-ray tomography, and this is the Mistral beamline where I'm working at ALBA, and also at Diamond B24 where Maria Arkeolaki and his team has also a soft X-ray tomography beamline. And with this, I thank you for your attention.